Hello, hi, a very happy good evening. So this is Krishna Ma'am and I'm talking about story of life on earth, and that's biology. So here we are with the board bio paper. So I hope you guys have done your bio paper really well because the paper was really simple. So I'm going to here discuss set 2 of your bio paper, and I hope you will enjoy the discussion because if you have got set 2, so you're really lucky. The paper was really easy. So but what matters is if your answer is complete or not, because when your answer is complete you tend to get full marks, but otherwise you might lose marks. So let's see how much you have written, because otherwise it's a very easy paper. So it will not take more than 15-20 minutes to discuss the paper, so it's going to be a very short discussion. Okay, all right, and today we'll close the chapter of both, so henceforth we are going to only talk about NEET. So from tomorrow I am starting your one-shot revision, so we'll be starting with the final round of revision. So I am starting with the living world, so you can stay tuned tomorrow at 5:30 to begin a final round of revision. All right. So here we go. So CBSE board exam. So we're talking about the bio paper which happened today. So as I dictated the rules, instructions on Saturday's class, that you have 3 sections. So totally you have to attempt 13 questions. So section A has 6 questions with 2 internal choices. So section B another set of 6 questions with internal choices, and then section C is 1 case-based question. Right, so we'll start with section A. The questions were pretty easy. So we start with this. So question A, so this figure was given, right? So observe the schematic representation given above and answer the following questions. You have to identify A and B. So we know with the given thing, so you have immigration. Immigration is to enter or come inside a particular population. So this is given an arrow towards the population. So similarly, something else should also add on to the population. So that is your natality. So natality is birth rate and mortality is death rate. Only these two are missing, but you have to write it in proper order. So the arrow is given towards the outer side because it decreases the population. So A is your mortality and B is your natality. So here the second part says, so this question carries 1 mark. If you written these two properly, A is this, B is this, you will get 1 mark, 0.5 for each. Okay. Calculate the growth rate of bacteria in a curd sample where 1 million bacteria increased to 2 million within a period of 1 hour. So how do you calculate? So the final growth minus the initial growth. So final growth was 2 million minus 1, so 1 million. So growth rate is growth per unit time. So 1 million per hour is the growth rate. So this is the simplest answer that you have to write, and this you get 1 mark. Okay, so you have an internal option for question 1. You can choose to answer the first one or this one. So here it is very easy. You have to identify the type of pyramid given below. So it is an expanding or a growing pyramid. On what basis did you identify? Because your pre-reproductive population is greater than the post-reproductive population. So that is the basis on which you observed this. Fine. So question 2, a boy developed some allergic reactions when he straight entered into his air-conditioned room after a game of football outside his house. Write any 2 symptoms that could be noticed in such condition. How does your body combat such a condition? So basically they are asking you symptoms of allergy. So you can write sneezing, coughing, irritation, watery eyes, all that can be written. So how does your body combat such a condition, meaning how does your body fight that condition? So as per your NCERT, for your allergy, you have your IgE fighting against during allergy, or your histamine and your serotonin are also produced. You can write that as well. So these 2 symptoms have 1 mark and this has 1 mark, right? So now we move to question 3. So question 3 again has an internal choice. So here the internal choice is given for question 1 and 3, right? So write the scientific name of the plant from where the natural cannabinoids are obtained. So it is obtained from Cannabis sativa. So you have to write it like this and underline. So mention the parts of the plant that are used for extracting this drug. So parts of the plant can be the flower tops, the dried leaves, etc. Right? So what effects does this drug have on the human body? So it affects your cardiovascular diseases. Sorry, it affects your cardiovascular system, right? So either you can talk about this, so this they would have divided, so this 2 marks is divided into 3, so this can be of, these 2 can be of 1 marks, or this can be of 2 marks, so this is the division it is given. So this entire, if all 3 are right, you get 2 marks, right? Yes? Okay. So epithelial lining, so it can be 0.5, 0.5 and 0.5, so it one can be greater than that, so it depends how they have divided the marks. So epithelial lining, so this or this, if you are attended this, well and good. So but if you do not remember the name of the plant, so well and fine. For all those who attended this, epithelial lining of our intestine is considered as the secondary lymphoidal organ. Justify the statement. Yes, the statement is true because you have your Peyer's patches, right, of the small intestine. So Peyer's patches of the small intestine act as a secondary lymphoidal organ because that is the site of interaction of antigens with the lymphocytes and it causes an effect. So that is all that you have to write in part 3. So coming to the next question, question 4, what are phlox? State their role in the biological treatment of sewage. So phlox are a combination of bacterial growth with your fungal filaments, right? So phlox usually come in the secondary sewage treatment. So phlox are aerobic bacteria, you can write, with bacteria, aerobic microbes. So they have bacterial plus fungal filaments. So what is the role in sewage water? So they decrease the organic content of the sewage matter by oxidizing the organic matter to produce the oxygen through BOD. If you like this, that is more than enough. So all the major terms should be mentioned. So question 5, how are the in situ and ex situ approaches of the biodiversity conservations different from each other? So here you have to write this only for 2 marks, right? So this can be for 1 mark and this can be for 1 mark. So the major difference between in situ and ex situ, this is conserved in their original place. So this is on the particular site in which they are endangered. So this is done on the site where they are endangered. So ex situ, they are taken off the original site and conserved in a different place. So you can write in situ conservation includes your hotspots, right? So you also have your national sanctuaries, your sacred groves, etc. So ex situ conservation includes your zoological parks, botanical gardens, etc. You can just give examples. So farmers are often suggested to use the following organisms in the cropland so as to improve the soil fertility, right? Example, rhizobium. So why should a farmer incorporate rhizobium in his farm? Because it fixes nitrogen in leguminous plants. So it is only for 2 marks, so this gets 1 mark and this gets 1 mark. So Anabaena is a type of cyanobacteria or your blue-green algae, and they add organic matter to the soil and they enrich the fertility of the soil, and they can also fix nitrogen. If all these points are there, then it is done. Okay. So with this we are done with section A. So section A was pretty easy. So if you have done if you have prepared really well, so out of the 6 questions, 6 2s are 12 marks, you should get at least 11.5, that is for sure, right? So now let's see section B. So what are the questions we have in section B? So restriction endonucleases are used in genetic engineering to form the recombinant DNA. Explain only with the help of the flow chart the steps carried in the formation of a recombinant DNA. Okay, so the only the flow chart is needed. Okay, so you have a foreign gene, so you have the vector DNA, so you use restriction enzyme, so you have fragments, So you use the same restriction enzyme, so you cut open the vector, right? So then you allow your gene of interest to come inside your particular vector, right? So you can draw and side by side write. So this carries, so here you have to mention the, suppose if you mention the terms like this, so you have to mention the site, okay, GATTC, sorry. So this is 5 prime to 3 prime, and then you write the reverse sequence. So you have to show the sticky ends, all that is important, and then show how your recombinant DNA molecule is formed, right? So question 8, write the inference drawn. So here you have 2 subdivisions if you see properly. So 2 subdivisions together carries 3 marks, right? So this can be 1.5, this can be 1.5. Write the inference drawn by Alexander von Humboldt after his extensive exploration of South American jungle. So he said, as we go deep into the area, the species richness increases, but only up to a certain area. The species richness is a rectangular hyperbola is your second conclusion. So now defining all the terms here, so S is your species richness and A is your area, right? So Z is the slope of the curve and C is your y-intercept. So this I have told you n number of times even when I was revising the sample paper, right? So I hope you have written this well. So question 9, you have A part and you have B part. So B part is in my second slide, okay? So given below is a stepwise schematic representation of process of gel electrophoresis. Identify the alphabets representing the anode M. So this is a gel electrophoresis. So your DNA moves from cathode to anode. Cathode is negative, anode is positive. So this is where I have the wells. So it has to ideally move from this end to this end. So this should be my anode, right? So this is S. The smallest or the lightest DNA strand in the matrix, so the smallest or the lightest DNA strand migrates very quickly. So that should be your R. So which one represents your agarose gel? Your T represents your agarose gel. So these are the answers that I want you to mention. So the second part, so that is 1.5, this can be 1.5. What is elution? Transfer of your gene of interest or your vector from the agarose gel into or you can write the purification of your gene of interest or vector from the agarose gel. So why is this step of particular importance? Because you have to dilute the agarose gel. To carry forward your recombinant DNA, you cannot use the agarose gel, right? So what is the elution step? So to cut the gene of interest or your vector from the gel, or you can write the transfer of gene of interest and the vector into the vial. So why it is done? So that you dissolve the components of the agarose gel, right? So we move forward. So question 10, again you have 2 parts. So it is 1.5 each, okay? So explain the property of contact inhibition and its effect on the normal human cell and cancerous cell. So first part is 0.5 marks, the first part is 0.75 and this part is 0.75, and this question we discussed it on the sample paper on Saturday. So what is the property of contact inhibition? When a cell grows in size and when it touches its neighboring cell, it stops growing. So it thinks that is its space, that is contact inhibition. What is its effect on normal cell? Normal cells follow contact inhibition, thus maintaining nucleus to the cytoplasmic ratio, whereas your cancerous cells do not have the property of contact inhibition. That is all you have to write. Why are biological modifiers like alpha interferon required for cancerous treatment? So they are actually how do they react to the disease? So alpha interferons are group of cytokines. They boost your immunity. So they boost your immune system and help your immune system fight against the cancer. So that is how they play a role. Fine. So coming to question 11, what are cryoproteins? With the help of a suitable example, explain how it acts as biological pesticide. So cryoproteins are a group of protein that produces the toxic insecticidal protein. So 𝐶𝑑�L means killing. So it kills the insect. Insecticidal proteins. So you will talk about how the Bacillus thuringiensis has this CRY gene in the inactive form. So, but when it is consumed by the insects, it gets activated to the alkaline pH of the insect's gut and it causes pros and thus killing the insect. Right. So, thus it can be used as a viable pesticide in cotton plants which is susceptible to a group of insects. So, that is how you write it completely. Fine. So, I guess this question is not tough. So, coming to the last question of your section B. So, you have A and B, you can either choose this or you can choose this. So, explain the concept of co-extinction by taking two examples. So, this co-extinction comes in your biodiversity. So, loss of biodiversity can be because of 4 ways. The last one is your co-extinction, right? So, when a host species is become extinct, so the predators that depend on the fish become extinct. Right. One more thing is plants and pollinators, because we have a mutualistic relation. So, when the plants die or become extinct, so does the pollinators. Right. So, this is done. So, forests provide intangible benefits to us, we agree. So, explain by taking 3 different areas how. So, the 3 different areas, again it's from the same chapter. So, it is narrowly utilitarian, broadly utilitarian, and it is ethical. So, narrowly, how it is useful to a single human, it gives firewoods, it gives medicines, spices, etc. Broadly utilitarian, it purifies the atmosphere, it stimulates rain, it holds your soil. Ethical, so we have to share the plant with millions of microbes, flora and fauna. So, actually each species is very, very viable and inevitable to us. So, that all comes under these 3 points. So, now coming to section A, this is case based question. If you have chosen this paragraph, you have to answer these 3 questions, and the next paragraph is in my next slide. So, here they have talked about the enzyme Taq polymerase. Okay, they have given the details. So, Taq polymerase, after its discovery, replaced E. coli DNA polymerase in PCR technique. So, today when I do PCR, I will use Taq polymerase, not the typical DNA polymerase. Explain giving reasons why the need felt for the change. Because in PCR, why do we need Taq polymerase? To do the extension step. So, in PCR, the temperature is very high. Our normal DNA polymerase can withstand only 25 to 37 degrees. If you take it from E. coli, it is 25 degrees. But for PCR, you need a much higher temperature in the range of 72 to 75 degrees. So, that is why we go for Taq polymerase, the temperature resistant. So, you need a thermostable one, that is the reason. So, what is a primer and it's important in PCR? So, primer is a short oligonucleotide sequence that is complementary to the template. So, what is its important in PCR? The Taq polymerase cannot initiate the reaction. Thus, we need primers. So, once primers grow and attach, your Taq polymerase can polymerize it. So, that is all the reason that you have to write. So, the next one, write the importance of PCR as a diagnostic tool. So, you have done RT-PCR yourself, so you should know, because it helps in identifying the gene by so it helps in identifying the host gene by amplifying it. So, that is the diagnostic tool. So, if you have not attended that passage, you have to attend this passage. So, this talks about your gene of interest, about your biotechnology. So, the question that follows are, name the human disease for which gene therapy was used for the first time. So, that is your ADA, adenosine deaminase deficiency. Explain the steps of gene therapy carried to cure the disease using the lymphocyte of the patient. So, you take a functional ADA, you put it in a retrovirus, put that in a lymphocyte, culture it and put it back into the patient. But it is not a permanent cure because lymphocytes are immortal cells, they have a limited lifespan. Okay, write the possible permanent cure of the disease by gene therapy that is in progress. So, the permanent cure can be when you insert the functional ADA gene at the embryonic stage. So, that can give you a permanent cure. Alright, yes. So, this is all the paper for today, and I don't think it was any tough, anything but easy, right? So, you agree with me? So for once and all, we are dusted with the CBSE for biology. So everything is so clear. So I genuinely feel even if you have started your preparation a month ago, you have still done wonders in bio. So it was not a tricky paper, unlike your term 1 was much tougher, I would say. So term 2 was pretty easier. So alright, so once done is well and good. So now we have to think forward. So we have to prepare for NEET. So tomorrow I am starting my one chart series as final round revision. So please join me tomorrow at 5:30 p.m. So we will start our round of revision. And this final round series will involve Menti quiz every Saturday. So Monday to Friday we will revise the chapters, and every Saturday we'll have quiz. So whatever chapters I have taught from Monday to Friday, those chapters will come in the quiz of Saturday. So this is our agenda for the upcoming NEET exam. Right? So alright, so stay tuned. So tomorrow at 5:30 I'll meet you. So until then, this is me signing off. So take care and bye. Thank you.